This last talk will be from Ireland, and we are proud to have the University of Galway and the CÚRAM, Centre for Research in Medical Devices, as members in our network. And Dr. Conor has a medical background in nephrology, but he also has a training in engineering, which is a very good combination to tackle the current topic of the clinical research as chance to develop new interesting medical devices. Dr. Judge, the stage is yours.
Thanks a million for staying to listen to my talk. So I'm presenting on behalf of CÚRAM, the Science Foundation Ireland Research Centre for Medical Devices. Give a quick overview of the CÚRAM Research Centre, some thoughts about artificial intelligence in clinical trials, and talk about a case study, SMART Trials, where we did a group sequential analysis using Microsoft in-database machine learning. We have a fantastic medical device industry in Ireland. 9 out of the 10 top companies have a base here. It employs nearly 40,000 people, and we've 12.6 billion euros of export per year. So second only to Germany. 400 plus companies are based here, and we have particular expertise in drug-eluting stents. So the vision of the CÚRAM Centre is to partner with industries to develop affordable, radically innovative and transformative device-based solutions for current unmet medical needs. The key to this approach is to establish unique networks of national and international collaborations, integrating world-class clinical, academic and industrial partners. So just looking at the pre-clinical area, artificial intelligence can help with prediction, so to accelerate the drug discovery, predict efficacy and safety based on the molecular structure for drug selection. Then focusing on the clinical trials, once again, it's all about predicting the sites, predicting the investigators and selecting them, finding undiagnosed patients to accelerate the diagnosis and screening stage, optimize the trials and study planning, predict the enrollment and screening, stratify the patient based on risk of disease progression, identifying subgroups, predicting patient persistency, and finally enabling adaptive clinical trials. And that's the case study that I'm going to talk about now. Adaptive clinical trials is where before the trial starts, you prospectively plan changes to that trial based on the accumulating study and data. This has to be done in a way to not undermine the integrity of the trial, to not increase your chance of a type 1 or a false positive. Most common type of adaptive clinical trial at the moment is a group sequential trial. We have planned interim analysis, for example, 6 months, 1 year, where you do your statistical test to see whether the trial has reached efficacy or futility based on a pre-specified stopping rule. The FDA has released a lot of guidance for both drug and medical device trials. And the FDA say that adaptive clinical trials have the potential to reduce resource requirements, decrease time to study completion, and increase the likelihood of study success. So this is supervised by Professor Martin O'Donnell, Professor Martin O'Halloran. Professor Martin O'Donnell is the director of the clinical research facility here in Galway, was PI on Interstroke, and prior to Galway worked in the PHRI at McMaster in Canada. Professor Martin O'Halloran is director of the Translational Medical Device Lab, ERC funded. So always when we think about a clinical question, we break it down into the PICO model, a population, intervention, control and outcome. So in this case, it was more of a technical question, but still breaking it down using PICO. P, the population being randomized controlled trials. I, the intervention, does automated continuous sequential analysis, compared to C, the control, compared to standard interim group sequential analysis, reduce the time from efficacy to futility for the primary trial outcome. So then we had to look at how was standard group sequential analysis done. As you all know, the data from clinical trials lives in clinical data management systems. And extracting data from those is a very resource-intensive task. For example, it might be exporting CSV files into your statistical environment. Some more sophisticated clinical data management systems might have an API for interrogating it. Then from once it's in the statistical software or SAS or Stata, the biostatisticians perform the group sequential analysis to see whether the trial has met the stopping rules for efficacy or futility. What we wanted to investigate is whether, instead of taking our data out of the clinical data management system into our statistical environment, could we bring the statistical environment into the clinical data management system? And that's where Microsoft Azure SQL Server 2017 came in. And this is where you can run your OR code and any analysis or packages available to OR, stored procedures inside the Microsoft SQL Server. So this cuts down on all of those steps of extracting the data and reduces this from a resource-intensive process to a minimal resource process, allowing, instead of it done every 6 months, to be done at any time interval, for example, every day. To retrospectively prove this, we used the SPRINT trial, which is an open access trial. Our group had previously done observational post-hoc research on this trial. It was a blood pressure target trial looking at blood pressure targets less than 120, the intensive arm, versus blood pressure targets less than 140, the standard arm. They had a composite cardiovascular outcome that showed a positive result for the intensive, the blood pressure less than 120 millimeters of mercury arm. The SPRINT trial used group sequential analysis, and their time from start to recruitment to time to efficacy was 5.67 years. They did their group sequential analysis every 6 months. Using our Smart Trials platform continuous sequential analysis, time from start to recruitment to efficacy was 2.78 years from start of recruitment. This is a saving of nearly 2 years. Stopping the trial for efficacy at this point would have yielded an estimated cost saving of 113 million, taking an estimated cost of 41,000 per patient, which was based on a survey published in JAMA Internal Medicine. Also, more importantly, it would have reduced 57 primary outcomes in the standard arm. But what's more, we proved that it was feasible to use continuous sequential analysis of clinical trial data directly in database using the ORIN database functions in SQL Server. So the impact of this platform is both ethical and financial. The ethical impact is avoiding exposure to futile treatments. The beneficial treatments are made available earlier. Financial impact is lower participant numbers, lower trial costs, and lower data monitoring committee costs. So that's it. Thanks for staying to hear me talk. I'd like to thank CÚRAM, the Translational Medical Advice Lab, the Clinical Research Facility, SFI, ICAT, NUIG,